Right, so let's start. Don't hesitate to interrupt if anything is wrong or if you have any questions during my talk. So hi again, I'm Guillaume, and today I will talk about semantic segmentation. So just before presenting anything, I just would like to take a small survey. It would be a bit interactive today, so for this session. So if you have would like to try a little bit the tool that I'll present today, feel free to follow along my presentation. Just to make a quick survey, I've made a book lab if you know this website. So if you can please make the so go on this website, booklab.com, and then you type the event code, and there is a survey just to so I'm asking just your level in Python and deep learning, so I can orientate the presentation depending on your skills. So you can be a very beginner, don't be afraid. You can know anything about don't know anything about programming at all. It would work perfectly. So I see that people are already answering. So right. So I see like 14% are people are connected. Great. So I just put here we go. So I already have 10 answers. Right, so we have already a rough idea of the audience. So basically, you know some basic Python. So great. Welcome. And so I skip on the certain very basic thing, but I will stay very basic on programming. So you will follow along. And so for those who would like to discover more complex features, I'll present the documentation and everything in this talk. So let's start. Okay, try again. So today I'll talk about Biom3d. So I'm doing like a short presentation, like 15 to 20 minutes, and then I'll switch to a demo, interactive demo, so everyone else can experiment with the software. So Biom3d is an easy to use and modular deep learning tool that I've developed during my PhD for 3D semantic segmentation. So to give you a little bit of context, this tool was developed in the collaboration between 3 different labs, 2 in biology and 1 in engineering, and we are interested in segmenting 3D images, so of microscope, either electron microscope images or confocal images. And basically what biologists are looking for is extracting significant measurable information, so such as the volume, surfaces, etc. And of course, some of you might know already that's one mandatory step, one required step to do that. You need to do segmentation, so which consists in extracting, in coloring, in labeling the region of the image you captured in here in certain color, but here you like to have what is called a mask, or black and white picture, where you have your white region, you have your object of interest in the right region. So here in red, the nucleus volume, and in here the chromocenters, so which are denser parts in the nucleus, so denser part of DNA. But segmentation is much broader than just microscopy, and it's applicable to medical imaging, also MRI, or any kind of 3D data. So in the beginning of this project, I've made a small survey on actually a quite significant survey on literature, what was the state of the art in this segmentation problem. And we actually found out that a lot of deep learning methods, so it was mainly deep learning methods, and a lot of deep learning methods were actually not reproducible. So among like 100 publications that we found, only 6 were actually providing just a minimum requirements, so which are the code, the documentation, the data sets, the deep learning environment, so which is the whole set of requirements such as a Python library required to actually use the tool. This raised the question of what is a good 3D segmentation methods, and one could answer this, a method that is performant for any 3D problem. So for example, I could segment anything from confocal images, electron microscope images to MRI here and CT scans with plenty of objects. So for example, here we have plenty of organs, but you can imagine the same with microscope images. For example, you could imagine, I don't know, mitochondria, cell membranes, you'd like to segment any organelles in the cells. So everything is applicable to medical and biological fields. Is such a method a good one? To a certain way, yes, and it's very challenging to do, to do a method that is actually flexible enough to be applicable to so many problems. And why is it even more challenging for deep learning methods is that basically deep learning method looks like this. So this is the interior of a plane, a Boeing 747, and this is what it looked like in the first version. So basically you required a full engineer during the flight, in addition to the pilot, to actually turn the buttons. And this is basically the first version of deep learning methods looked like. But good news, this is the current version of the same plane, and it was actually realized also for deep learning methods in a certain way, and also for 3D segmentation methods. But so yes, it is a good segmentation method that is able to do all of that is good, but is it enough to be universal? And unfortunately, no, it's not sufficient, because when you actually build a method that is able to do this, so to segment a lot of problems, you actually create a sort of an interface, usually a programming interface. So for those who know how to use command line, they're aware of this kind of interfaces. And so you actually satisfied a quite short range spectrum of users, because on the other hand, especially when you are doing microscope image analysis, you would like to satisfy also biologists, which are users who expect very simple interfaces, like just a simple lever, 2 buttons, and something that is able to do what they want, which is you're fine, but in the deep learning world, which is segmentation here. And so without any need of complex configuration, but they still want some fine, they could access some fine parameterization. And on the other hand, you actually need also to satisfy deep learning developers, because if when you open the lid, when you open the engine of your deep learning, it looks like very messy and everything is entangled and you cannot change anything else, even though it looks very nice from the outside, your community will not take care of the code and will not maintain everything. So your method will not be used. So basically what you would like to do to have a universal method is to satisfy all of these profiles at the same time. And unfortunately, most of the methods are satisfying only one specific profile. So yeah, just here roughly represented what I've just said. So basically here we have our 3 user profiles depending on their need and the skills, and on the y-axis we have the estimated time they require to actually take these methods, to use these methods. And so on this end, on the left end, we have the beginner who don't have any graphical user interface, so they're stuck and they need to learn how to program. And so basically they need a lot of time. It's not possible. And on the other end, you actually have developers that are facing complex code, no documentation, and a poor code modularity. So code modularity is a key importance in this world, because if you want to plug in or out new pieces of code, so in deep learning, it's moving very, very fast. So you constantly want to change the model. If it's not easy to do, it will be very difficult to maintain for the developers. And so they would give up and have to rebuild everything. So the goal is that, and this is what is a universal method. And so Biom3d is an attempt to do that. So basically to have a method that is available that can solve any 3D problem on one side, and that can address the expectation of a broad spectrum of users. And that's what we talk about today. So for the non-programmer side, we have 3 different interfaces. Today I'll present 2 of them. So the local one, which is this one, which is basically the one that you have when you install the software, when you just launch it. And so I will go very quickly on that. Here I take advantage of this interface to just explain you how deep learning quickly work. So you have as inputs to train a model, to train a deep learning model. So you have to train them. You need 2 folders. One folder containing raw images, so your images you capture with your microscope, and annotated images. So these images are done manually with tools like NaPari, and I saw that in I2K UAD presentation about NaPari. For instance, it could be anything else, but you need these masks, so these black and white pictures or grayscale level pictures that represent your object of interest. You don't need for 3D images, it works usually with below 100 pairs of images. So often people ask how many images is required to have a good training data set. So really depend on the difficulty of your problems. But as an experiment, so far, I've found that with less, around 100 images, or it works fine. So, and for nuclei, for example, in problems which is quite simple, you need less than that. So that's your inputs. And basically, with Biom3d graphical interface, you could just click on auto configure, and the software will take care of the configuration, so which is the pre-processing of the data sets, and configure the training configuration file. So the size of the model, the size of the patches, because images in 3D should be patched, etc. So many parameters automatically taken care of, and then you can start the training. So once the training is done, an export is made, which contains the trained model. And then there you go. So you can now use it on raw images, so images that are not annotated, and you can use them to, I don't know, make prediction on 100 or 1,000 images without automatically. So you need 2 things: your raw data set and your trained model, which is was done just at this step, and then you get your predictions. So it's as simple as that. And today I also talk about the collab interface of Biom3d, because it's actually a bit more handy, because it doesn't require GPU or tricky installation. Biom3d is a package, a Python package. So it's just simple as installing a package, but as it relies on PyTorch, which is a deep learning framework, you have to install CUDA and PyTorch. And sometimes, depending on your computer, it can be a bit tricky, but it's doable. It's just something it can take time. So today I also present to you the Google Colab version. Here it is. Just quick snapshot, and I present the detail after. About the Python programmer and also the non-programmer, I also created a documentation. So here you find tutorials, and so everything that I present today, you also find it on the documentation. And also I've detailed the different functions. So this is more addressed to also a deep learning developer, so they can actually delve into digging into the code functionalities. We set ourselves the expectations of reaching the state of the art. So Biom3d works on a plenty of, but actually all of these images that I've already presented are predictions from Biom3d. So it works on plenty of different problems. And finally, I have so Biom3d is a modular, which means that this is, for example, I just represented here the structure of a code, the typical code that actually I found that was actually able to do 3D segmentation on different kind of problems. Every little dot represents one Python script, and every link represents how Python scripts are calling each other. So if you want to change anything in that, you actually need to understand every links and how that works and how it interact and replace everything. So it's very complicated. And this is how Biom3d is structured. And both codes here are actually reaching the same accuracy. And another big importance with modularity, as I already mentioned, is that you can actually, it's not limited to 3D segmentation. Actually, Biom3d is not limited to 3D segmentation. I've already done classification. I've done self-supervised learning, nothing like that. And you actually don't need very specific knowledge. You just need to change a configuration file from one model to another. And then you can keep your graphical interface as it was, still using the same one, and change the model inside. And just for a quick example, for example, I just segment a data set, pancreas data set with different models, and I've reached a better accuracy because I just changed the model. So which I was able to do thanks to this modular interface. Right, so demo time. So before the demo, if some of you would like to try on their own data sets, feel free if you already have annotations. If not, I propose you to download a data set that we made in our team. So this data set is a data set of a nucleus. You have 39 images. And they are annotated. The nucleus or the chromocenters are annotated. So today we will do one of these tasks. But you feel free, if you want to try at home, then to do the other one. And again, if you have, if you would like to do annotations, I really recommend to use Napari. Let's see, there's a. Oh yeah, for the link, Giovanni, for the data set, I send it in a few seconds. And just before that, so for the data sets, you'll find the link on the Woo Club. I just changed it right now. Here we go. So you should find the link on the data set, and you should be able to download it. It's not that heavy, should be 80-ish megabytes. So so if you can go to Woo Club for those who just arrived and go and type this event code in your event code research bar, and so download the data set. I actually don't really know how, if everyone else, if you have any problem in the downloading the data set, you can just send a message in the chat. And once you downloaded it, you can unzip the file, and you should find 4 different subfolders. Oh yeah, thank you for sharing the Google Clap link. So for those who didn't see, Giovanni just shared the Google Clap link in the chat. So you can once you have downloaded the data set, you can unzip the file, and you should find inside 4 different folders. So in the first one, it contains the different raw images, which are the nucleus images. The second one contains the masks, so the segmentation that were done manually, and I'll show you how they look like. And the second one are the chromocentrous masks. The last one is a set of nuclei that were not annotated, which is there is 120 nuclei. And this is the goal. So the goal is to be able to actually make predictions for this folder. So this is the folder of segmentation. So I show you here, I have NumPy open. Let's hide everything. So this is one of the sample in the data set. Here you have one nucleus on the side. We are not interested in that, but we'd like just to segment the one in the middle of this image. And you can clearly see the different chromocenters, which are the denser part of the nucleus. So the biologists in the team that is involved in this project, they segmented the nucleus. So here it is. And they also segmented the chromocentre. So just an example. If any of you has a data set that are curious to use, they could also use their own data sets. And just as a quick, just to show you quickly how it was made, basically what they did, if I just take this one, they take the brush here in Avery, just create a new layer, and then you can just paint like that. And this is, and you can go to the next slide. Right. So erase this. Here we go. And so if everybody else has downloaded the data set, we can now go start the experimenting with the tool. So first of all, I would like to ask you one question. So would you feel more comfortable trying the local interface or the Google Colab interface? So the local interface, there is just I would share the link and put it in the chat in case of you don't have it already. So the difference between the two, the local interface, you have unlimited GPU because it's run on your computer and your data stay on your computer, so offline. But you need a GPU to run a training. But a small one works with these data sets, and you will require Anaconda and a little bit of work to install it. It's 4 steps, not that much, but still. Or we use Colab today, and there's no requirement for any GPU, no installation required, and you have you just have a limited time of GPU, but and we require the data to be uploaded on your Google Drive. But it's really easy, really handy for this demo. The only thing is that once you're at home, you would probably prefer the local interface. And there is also an option for both, because you can also train a model on the Google Colab, then download it on your computer and make predictions then to train on your computer with your trained model. But it's a little bit denser program. So I'll let you decide what you prefer, and we'll do one. Look like I see that there are 20 people. Don't be shy to answer the call, the survey. All right, so I can already tell it's maybe it will change, but for now the dominant answer is Google Colab, which will be fine, and I present the rest a bit. The rest of the presentation can be a bit shorter about the rest, if we have a little bit of time in the end. Okay, 1 or 2 minutes, maybe 1. Right, so it seems that we are decided. So Google Colab it is. So for the local installation, those who are interested in using these installations, so I can advise you to, recommend you to look at the documentation here, just to go through the installation and see if you have already an Anaconda. You create an environment, you install PyTorch with 1 line, and Biom3d with a 2nd line. That's it. It's very, very short. So PyTorch is quite heavy to install, but Biom3d is really, really small, so it shouldn't take more than 5 or 10 minutes. I can also put the link on the chat again. It's a good thing to do. And you find the code also here, which is just a general all of the methods. And about the Google Colab setup, so what you need is a Google account, then to unzip and upload your training datasets and the prediction datasets on your Google Drive. So basically what you can do is my drive, so I have my drive here, have data folder. I have already plenty of things, but what you can just do is unzip the file and just put it like this on your Google Drive. Drag and drop it. And when it's done, I have it here. Yeah, I have 3 folder here because I put the prediction one in a separate one here, but don't worry about that. And so as it uploads on your Google Drive, I'll present you the Google Colab. So I copy the link and put it on the chat, and also you have it on Google. You can just click on that and it opens. And here we go. Maybe I can zoom in a little bit. Right, so you can see it. I hope that you can still see my screen with the Google Colab. I think I'm sharing the whole screen. If you have any problem with the dataset download or clicking on the link, something like that, just feel free to ask or to tell, or to upload your dataset on your Google Drive. And of course, if you have your own data, don't hesitate to ask me if you have any problem or anything. So first of all, once you're connected, so you must be connected with your accounts. And once you're connected, you can click on connect here in the top right corner. And it's connecting. Here we go. So once you're connected, you have to make sure that you are connected to a GPU instance. And to do so, you can just I've just added a small script here. Okay, you have to trust me. I'm not going to pirate your computer. So you can just run this first block, and here it will indicate you the type of GPU you have and if there is any GPU. So here I have a Tesla 4. And if it doesn't work, you can just go on runtime, change runtime, and then you can use T4. Right. Once you've done that, what I can do so is because I don't have any feedback from you. You can raise a hand, I think, on these. Can you raise hand when you're ready to start on Zoom, so I can see if everybody else is ready? Or I think, let me see. Yeah, okay. I can see that several people are already ready. Only Alejandro is ready. Miriam. So for those who didn't catch up, what you're trying to do here is use Biom3d, so segment with Biom3d on this Google Colab, using the link in the chats that you can find on Zoom, and you can find also in the WooClap event here, and the link is also available in the chats of the Zoom call. If you have any problem, don't hesitate to ask. All right, so let's carry on. For those already, you can start installing Biom3d. So by just clicking the arrow here, and it will install everything. Shouldn't take very much time. And when it's done, here we go. you can mount your Google Drive, which means that you connect your Google Drive to You connect to Google Drive to this notebook. Hello. And you will have access to your Google Drive data. So let me just move this. So your Google Drive data that are uploaded. Here we go, it's mounted, and here you can click on File. You have the green arrow here. G Drive, My Drive. And here you find your drive. So in my case, it's in the folder called Data. And I have my data sets here. So for now, we just leave it here. We just check that I can access the data set. So again, if you are not able to access to the data set, just don't hesitate to ask. You don't see the chat anymore. Chat. Here we go. Right. So now let's go to the to the rest of it. So we would like to first precise a folder where the model will be exported. So the Biom3d model. Here in my case, I've just created the model folder here. And if you want to create a folder, you can just click on the 3 dots here, new folder, and you can create a folder like that in your Google Drive. So this folder will store the deep learning model when it's finished, and as well as the configuration files. And I'll briefly explain to you what it is. So once you've created or decided which folder you would like to store everything else, you just click on the 3 dots, copy path, and you paste it in this field. You can now define a fancy name for your model. So here it would be that, I don't know, nucleus, could be anything else, could be the default, works. And here you can put the 2 folders that I was talking about. So the first one is the raw image folder. So let me see, here it is. So this is the image folder. So I can copy path again and paste it here. And once it's done, you can also do the same this time with the mask folder. So here is up to you. If you would like to segment a nucleus, you can choose the first one, masked nucleus. And if you'd like to segment chromocenters, I'll let you decide. You can choose the second one. I'll go for the nucleus. And once it's done, you can choose. So here is the number of classes. So the number of classes is the number of objects you have in your images. In our case, we only have 1 class, so the number of type of objects. So in both cases, we have either just 1 nucleus or several chromocenters, which is 1 type of object. It can be in your images. You could have, like, for example, segmented in the same image the nucleus and the chromocenters. And in an APARI, you can do that by just by adding a label, for example. You can have to just segment the nucleus like that, change the label here, and annotate the chromocenter. And here you have 2 labels. This is the number of classes or label that you have. So here we have only 1, and we stick with 2 epochs. So epochs is the number of time basically that your model will see the entire data sets. Here we'll do it twice only. It's a bit too small, too short to have something very, very good. Usually 100 is recommended. And if you want to have something almost perfect, you go for thousands. But 1,000 is only available on your offline computer, because it would probably require more than 6 hours, because on Google Colab you're limited to 6 hours. So once you're ready, you click on the arrow here. And again, if you have any questions about that, just feel free to ask. And here we go to the pre-processing step. So this step is essential. So basically there are several things that are done behind this piece of code. First, your data is normalized and is also checked. So for example, if you have, let's say, several channels, like for example, you know, in microscopy, you can have several images with several channels. And in certain cases, you have 3, and in other 2, this piece of code, we say, okay, we raise a warning and say, careful, some images have 2, other are 3. Or for example, if you have in certain cases only 1 labeled, and in another one you have 2, which is actually possible. For example, in our nucleus chromocenter case, we can have images with no chromocenters. And this also we say warning some images, and the images is indicated as only 1 name. And if you actually, this is the determinant step. If you have the green arrow here, you're actually good to go with your images. I try to actually make sure that plenty of cases are taken care of. Like if your channel, for example, in 3D images, you have different order. Like you have in your dimension of your image, you have height, depth, width, depth, and channel. And sometimes you have the channel in the beginning. So I also try to take care of that, and many of things like this, many of cases. So if you have any problem with that, you just feel free to ask. And I'll give you my email in the end. So if you try and you have a problem, just send me an email. And now we can start the training. And here we go. So the training started with 32 images, and 7 images were used for validation. So just to give you a bit of more detail. So here what is plots is the nnU-Net architecture. I won't go through all the layers, but this is the deep learning model that is currently running. And what is displayed here is the loss function. So the loss function is the function that gives you the accuracy of your model. And basically, the closer it is to -1, the better it is. So currently, when we see it decreasing, it's a good sign that we are on the right track. So currently, it's decreasing. And we actually have 250 steps in 1 epoch, and we would go through 2 epochs. So it shouldn't take more than 2 or 3 minutes. And on the nucleus problem is almost sufficient to have something already quite good. And I'll show you later. So another thing is during the pre-processing, I told you that it was doing 2 things. First is the normalization and the sanity check, I would say, on your data set. And the second one is the configuration of your training model, And so the configuration is stored in 1 single file. It's a Python file for Python user. It's a configuration file that was written in Python, so it's a bit easier to read for those who are used to it. And you can find it in a config folder that should have been created in your Biom3d folder. And in my case, so we have the date here, and I can just double click on that, display it. And here it is. So this configuration file, I've tried to write a lot of comments on every configuration, everything. So this file was generated automatically depending on your data set. For example, here they detected that there is only one channel in the image, there is one class. I mean, one class was precisely you, but the channel axis is the first one, the thing that I was talking about. And for example, here you also have the configuration of how the data will be pre-processed to be passed to the nnU-Net model. So for example, the images should be patched, so cropped randomly in the image. And this is the ideal patch size that was found by Biom3d. Right, so everything in this file is editable. So if you change anything in there, you can actually start a new training. For example, let's say you think that this patch size is a little bit too small. So you can change it. Oops, I can increase it, for example. And then here I have, so I save it. And when it's saved, I can just copy the path here and start the training again here directly. And you won't have to go through the pre-processing again, because your data is already pre-processed. The path of your data is already precisely here mentioned in your configuration file. So you don't have to go to the pre-processing. You can start directly from here. And that's what I wrote here. You can just have a custom configuration path. You just copy it here and you start the training again. So I won't do that today. I won't do that, but you can actually experiment with that. And here we go. So the training was finished. It's finished now. Training done. And we now have a ready-to-use model. So in your folder that you define again, you have a model, logs folder, sorry, a log folder. And in this log folder, I can find today's model. And I can see that in the submodel here, I have already trained models. I have nice images. I cannot see anything, but I have images explaining, showing what the model is able of. So for example, you can see that here there is a mistake because the ground truth is not perfectly predicted, but I cannot see on the second image anything else because it's not on the right stacks, but it's all right. So this model might need a little bit of improvement to be perfect, but it's actually already usable. So you can actually use this model to run some prediction. So even if you quit the page, even if you stop everything, you can still restart reopening this link, and as is it stored on your Google Drive, you can just enter your log path, which is this path here, the path of your model, and copy it here, paste it here, and then you can enter a prediction folder, so a folder like you'd like to predict on, so raw images. And if you have like me already uploaded the folder that is called row selection, etc. So it should be in one of the 4 folders in your in the data set that you downloaded. You can copy this path. So this folder contained the 120 images. Copy path and paste it here. And this last field is where you would like to store the results. And I've created already a folder. So you, in your case, if you want, you can just create a new folder. And when it's done, you can just put the link of this new folder. Copy path here. And you can start prediction. You can start predicting images. And here we go. So it's quite fast. And For those who were interested in the local installation, so using Biom3d on your computer, you can actually download the model here, this folder, and you can use it into Biom3d in the local installation. So as you go through the prediction process, we can already see the result of the model in the prediction folder that I just created. So here you can see that a new subfolder has been created with the name of your model, and the prediction are appearing. So I can just download, for example, one, and I can download the raw image to predict just the corresponding one, just to see if everything is correct. Download. Here we go. So I have my two images. I can go back to Napari with everything else. Open my download folder, and I have my two images here. One should be the prediction, so this is the raw image. And in Napari, if you want to convert something that was predicted, you can convert it to a label like that, and you can switch to 3D. And here we go. So we have our nucleus. So you see this one is quite complicated. It's a quite complicated nucleus. You have a big hole in the middle. You have noises around it, and the model actually successfully didn't take these noises into account, which you wouldn't be able to do with a classical tool, which are non-deep learning tools. Deep learning is able to actually, for example, if you just use thresholding in this case, you won't be able to make it. You have to do some post-processing to take care of all the thing. And here it was done in less than 20 minutes. You are ready to use model. Right. So I don't know if some of you were able to go through all of the process. If you have any questions, feel free to ask. Otherwise, if some are interested, I can go through the local interface also. Did I lose everyone? Maybe. If some of you are stuck to one step or stuck to the first step, you're just unable to determine. Oh, thank you. Content. I think what you've done. Yeah, I think what you've done is Li-Yuan, what you've done is that you've passed the parent folder. You did not pass this row selection. If just
I see. Sorry.
Yeah, I think you passed just this folder.
Got it.
Yeah. Great. Awesome. At least two of you are able to do some predictions. Oh, that's great. Well, if you wanna go through all of the process, you can just let it go, but you can just interrupt it and restart it when you want. You don't have to go through the whole prediction process. But yeah, now you are able to do to train a deep learning model with 3D images. So as we still have a little bit of time, okay, first, any other questions in the audience if you are? We just come back on the WooC lab. I think I've noticed that at least 6 of you were actually interested in the local installation. So as we have a bit of time, I can also show you this local installation. And when it's done, you can actually download the model that was trained on Google Colab. If I can display it, here we go. And use it on your own laptop. Oh, all right, Alejandro. Bye. Can I train on small volume and predict on larger ones? That's an excellent question. Yes, you can do that. As long as because the patch size, as I said, the model is actually patching. This method is actually patching the image. So you can train on small volume because then the model will train on will predict big patches in the big image. So it works. And just to be sure, you have to, in your small volume, you have to annotate everything. So if some of the region must be ignored, just don't annotate them. And if some of them, so be sure to annotate every region that must be labeled and not say, okay, I'm just annotating the middle of my image. And so better crop the image. So that's another reason why it's good practice to actually train on small volume and then predict on big one, because then you can just segment the small volume and everything in this volume and then predict on the big one. So it's a very interesting question. Yes, Alex, it includes DICOM images. It works with DICOM images. I have not tested it, so because I'm using a package called Simple ITK, and so normally it detects automatically the format of the image, and I think I've read in their documentation that they work with DICOM, but test it, and if you find that it doesn't work, just email me. I've tested, for example, Nifty format a lot, and it worked really well, but DICOM should work too. Right, so just to quickly show you you're welcome, just to quickly show you the local interface. Here I have an Anaconda prompt. So for those who are curious to install it, I can just show you. So you have the documentation. I already, I think, shared with you the link of the documentation. Yeah. So I can share it again in case. And so in this documentation, you can find how to install it and how to use it. So here, how to use the graphical user interface and how to install it. So Biom3d is a Python package. So it can be installed as a Python package. So it's quite easy. You can just pip install, I don't know if it's pip install Biom3d. But first, it is recommended to create an environment. So for those who know, there is Anaconda, which is environment manager. So basically, it's take care of isolating all of the function, all of the libraries that were installed on the computer. So you can install that, and when it's done, you can start an Anaconda prompt. which looks like this. Basically, you look into your Windows installer in research bar and you can find it. You create a new environment with these commands. You can copy and paste, and then you can install, activate these environments, install PyTorch, which is the deep learning library, so the set of functions that Biom3d rely on, and then install Biom3d. And when it's done, you're ready to go. So with only one line, for example, I show you here, I can start the graphical user interface of Biom3d. And don't be scared, you find everything else in the documentation website. So let it start. It's a bit long. And I can, in the same time, I can see that it's finished already, the 120 images. So we don't need it anymore. Just a final point about this notebook. You find on the last block, evaluate your model. So for example, if you have a test set, so which is a set like the training set, but you did not use it, you isolate these images during the training. So you have a set of raw images and there are annotations. And these were not used during the training. You can evaluate your model. So the precision, the accuracy of your model with these. It's a quality control, and it's very important in certain cases. If you want to be sure to measure the accuracy of your model, you can use this block. Right. But I was looking at that because I wanted to download the model. So if it allows me, I can go back to my Google Drive, go to my Biom3d folder, and here I can find the log folder and download the last one, so the one that was created today. I can download it. There is an arrow here, it's zipping it. And as it's downloading, I can show you Biom3d interface. So sorry, it's a bit small. I don't think I can zoom on this graphic, on this interface. Basically, if you cannot see it, there is two options. So Biom3d is made also for biologists who don't have access to a GPU on their laptop, but they have, for example, I don't know, like a data center in the lab or a server where there is computational power. For example, microscope server, they have like a larger computational capabilities. And what you can do is connect to this server. So if you have your server address, you use your username and etc. You can remotely connect to a server with a GPU and then use it. But if you don't, and if you would like to use it offline, so on your laptop, you can just click here, start locally. And here you have just two tabs: pre-process and train, which is exactly like what we had, sorry, in the online version, and predict, exactly the same. So today I'll show you the predict one, and one important information is that Biom3d is compatible with Homero. So for those who don't know, Homero is I would say that a data manager. So for microscopic images, let me just show you. I have one account here. I just present you how it looks like. So Homero is a data manager. So for example, if I have here some images. Let's see if I have the raw images. Here we go. So this is how it looks like, and you can store your microscopic images. And it's a server, so it's a server client version. So here I'm running it online, but these data are stored on the server. And if you have this kind of tool in your lab, Biom3d is compatible with it, and you can actually enter the dataset ID and run a prediction on this dataset without the need to actually download the dataset on your computer and put it back on Omero server. Right. So just a small parenthesis. But in our case, we have our images offline, so on our computer. And I have it. So I just downloaded the model here. Let's see, can extract it. So this is the downloaded model from my Google Drive. Here it is. I can find the different subfolder here, and I need the data sets. So this is the data set. Here we go. So I go back to Biom3d interface, and in the first field here, it indicates select a folder containing images for prediction. So this is this folder, the folder containing your images that are unannotated. I just copy paste here, and I do the same with here, the model selection. So this is the folder containing your model. And when it's done, you can send either the prediction back to Omero for Omero user, or you can just keep it here on your computer and you select a path. I don't know, just create. Let's say, create a new folder, prediction. And I can just put this path here. And one of the other advantage of the local interface is that you have some post-processing that are included into the local interface. For example, you have the option here to just keep the biggest object in your image. And in our case, it's very interesting because we would like just to keep the nucleus, and we'd like to remove, sorry, all the noises that could surround the nucleus in case it exists. Another option is also to keep only the biggest object. So this one actually selects automatically the object that are the biggest one in the image, and remove the small tiny noises around the large objects. So here I just want the biggest one, and I can just start here, click on the start button, and I have a terminal that is open. And here I can see that it's running. So I just start the prediction, and it's exactly the same information that is displayed on the on the Google Colab. So it's running. As I said, here I don't have a big GPU, so I don't even have a GPU. I think it's very, very slow. You see, much slower, like 26.7, much slower than you have on Google Colab here, 1 iteration per second against 19 seconds per iteration. So very much, like 20 times more slower. But well, if you're okay with that, you can run it all night, and you have your prediction in the morning the next day. So it works also. Right. That was it. If some of you would like to, if you have any questions, again, don't hesitate. And if you would like to go through the installation process or anything else, so if you have any question about the rest of Biom3d, we still have a bit of time now, so feel free to ask. Not sure I can see your hand if you raise them. I will stop it because it's Do we need to have Omero installed on the computer for the offline version? No, you don't need to have Omero installed. it's actually, so for those who are interested in Homero, there is in the installation process, one tiny thing to add is to install, you need the Python package of Homero to use it. So there is one additional line to install. But that's a good question. So I'll just interrupt it. Here we go. And yeah, just to finish, I can find the prediction in this folder and saying again, this was predicted on my computer this time. Thank you very much. I did not share my email address. If anybody else is curious to want to give me feedback, I can just put it here. Did you try out other architecture or only nnU-Nets? I tried other architectures. So I've tried EfficientNet, for example. So I've actually made, I have to recode it so to be adapted to different patches size. But I've tried it. It works better. So it's it reaches a better accuracy, but to the costs of being slower. So you reach 1 or 2%, you have 1 or 2% higher accuracy, but you need 1.5 times more. Like, for example, I can give you an exact, like in my presentation, I've run some figures here. Yeah, for example, here I've tried an EfficientNet and I've increased the accuracy by a bit less than 1%. And I've reached 16 hours. I needed 16 hours to train this model. So currently, I don't have a better trade-off than the one that is the default one. But if you, as I said, as it's modular, if you are curious to try different architecture, you'll be able to do it with Biom3d. So just to show you, I actually, so oh yeah, I have suggestions. Yeah, my this email address, I've just realized that this email address is actually might be unavailable in the upcoming year because it's my previous email address for my previous lab. So I just share it here, the one that is here, and this one should be available for forever, hopefully. Yeah, about the I want to say, yeah, about the configuration file, I also want to show you here. We can open it. You have a model here, which is the default U-Net model. And if you go to Biom3d, you have actually different, no, it's not available here. You have different, you have a register with different options, and you can change this model to another one. This is where, without having to code anything else, you just change the configurations. For example, this one is the one that is close to the nnU-Net one, but it can be another model. If you have a transformer model, I would try between transformer models, for example, if you know, and also worked. You're welcome. Right. So if you don't have any other questions, I can just advise you to try it out if you have 3D segmentation to perform to do, and really don't hesitate. So it's still in development, so it's already been tested by 2 different teams in our lab. It's even 3 different teams. Sorry, it works on different images, even like on membranes or things like that. I mean, a bit more complicated stuff. It works. So if you like to try and it doesn't work, we actually will be really very curious to hear from you and to give. So don't hesitate to give me feedbacks, and I can improve the software. That's how I included all the cases that is in the nature. Right. So if you don't have any more questions, I just have to wish you a good luck for your next experimentation and nice sessions for the upcoming ones if you assist to the rest of the I2K events.